But this is actually very relevant that we have met the enemy and he is us, because what is happening right here, very appropriate, after the viral pathogenesis, what we mean many times after the viral infection, our own immune system create all these problems. And we are going to see that. And that's the reason that we have met the enemy, that's the virus, and us is our own immune system, which we will see that often cause severe damage and even death. So basically us killing us sometimes. So I just put a long introduction in the front slide. You will be able to see, and you just read through that. There is really not much that the question wise that we are going to ask, but it's very relevant to understand that viral pathogenesis um, in this current context as well. So just some specific definitions: that the pathogenesis capacity of the pathogen to cause a disease in a host. And we understand this by the signs and the symptoms. And the difference in between that the signs are these are usually caused by the immune response, whereas the individual difference is that signs at what other can detect. So for example, if you have a fever, I can take your temperature and I know that more than 98.4, you have a fever. I can detect that. But the symptom is a headache. Someone else cannot tell that you have a headache. Like that's a problem. The symptom that you can feel, but others actually can't. If you have a rash or anything reddening, those are the same signs that can be felt, but not the symptoms. On the other hand, the next other definition that you may have already seen in your microbiology class is the virulence. The virulence is the severity or degree of the pathogenesis. So pathogenesis is the capacity of a pathogen. Here in the virology course, it's a virus, but it could be any pathogen to cause a disease. And how bad is the disease is the virulence. And this can be measured and quantitated as well. And one initial thing that the viruses can be virulent. We know that what is a virulent virus could be an avirulent. We have in the very first chapter, we have known that there are many viruses which actually pass through us. Some of them can get into our cell even and stay there without causing any damage. On the other hand, there are also some which are attenuated, and these are the viruses which accumulated some mutations and lost the ability to surmount an infection, productive infection, but they still can replicate in some of the cells, highly used by the vaccines as vaccine strains throughout. So how we quantify the virulence, we can just look at some of these parameters like in incubation time for the appearance of the signs. We can measure the virus titer. You know that how to calculate the PFU, plaque forming unit, a multiplicity of infection. You all know these, that how it can be done. Mean time of death of the subject, that's important. You can measure the fever, weight loss, pathological lesions that you can see just like a microscopic slide, like a histological slide under the microscope. Also, you can count some of the blood cells, which is very relevant to HIV-1 because it infects the CD4 lymphocyte or the CD4 T cells, which is the T helper cells. And these helper T cells numbers actually do reduce. And that's the reason that we are going to see that these are the helper T cells. They are going to awake your acquired or adaptive immune system. If you kill these cells, you do not have anything to call your adaptive immunity. And that's the reason that the HIV becomes so dangerous, a reduction of CD4 lymphocyte. And again, we are going to use these names all throughout. Remember that the CD4 lymphocytes or the T cells, also CD4 T cells, are the T helper cells. Remember that. And the T helper cells could be Th1 type, Th2 type, T helper 1 and T helper 2. We have seen that. And there will be another type of T cell, which is a CD8 plus T cell or CD8 T cell, which are the cytotoxic T cell. When they are engaged or presented with an antigen, they will look for the virus infected cells and do the killing. CD4 help in awakening the immune system, whereas the CD8 or the cytotoxic T cell will kill the cell. So just remember that because we are going to use this term CD4, CD8 throughout this chapter. All right, so first we are looking at the incubation period. You do not have to remember any of these, but just shows Let's say, for an example, influenza rhinovirus, you have an incubation period of 1 to 2 days or 1 to 3 days. In the middle part, say, for an example, measles, smallpox, or say take a chicken pox. Say, for example, 13 to 17 days, which is just about 2 weeks is the incubation period, which is pretty long. On the other hand, you can see that, say, for an example, rabies, it could be 3 months. Papilloma virus, it could be even more than that. So the appearance of the disease takes a long time. So here is a word that we have used, and you may have heard this word in your microbiology class, is the prodromal syndrome. And I just mentioned that what prodromal syndrome means or symptom means. Prodromal is the vague onset of a disease before the acute disease. As I said, that when before getting a cold, you say a deep day before you really get the cold, that I'm coming down with the something, you feel itchy eyes, throat, and just don't feel all right. That vague symptom is called the prodromal symptom. And that is the period. First, you get the infection, and from that, the appearance of the prodromal symptom is the incubation period. All right, so how do we study the viral pathogenesis? Is there are lots of limitations. We can do that in the cell culture. We have lots of animal models like mouse, in the macaques, monkeys, many other animal models as well. But the problem that you can easily see that there are lots of problems. Problems means that in the cell culture, it doesn't have an immune system. And the viruses can replicate in some of those cells. But when you put that viruses in an animal or even in the human beings, you cannot replicate those results. So it's very, very different. And that's the reason it is very difficult to sometimes quantitate these functions. Say, for an example, this is I just wrote carefully understand the rationale of the experiment. And this is a very classic experiment that was done. So for the influenza virus, the sialic acid is the receptor. And you have seen that in the previous slide in the virus entry chapter, that a sialic acid is bound to a galactose, which is the second position. So this is the tip of that glycoprotein, sialic acid. And this is bound to the galactose by an alpha-2,6 linkage. And when these were made and tested in the cell culture, they have found the virus actually bound to those sugar residues. And it was determined that the sialic acid galactose, or the sialic acid, is basically the receptor for that. It was discovered by that way. Now, when the experiment was done in a mice, which was where you have the ST6-GAL1 gene knocked out. So this ST6-GAL1 gene does produce this alpha-2,6 sialic acid galactose. So if you knock out these gene, the glycoproteins in the respiratory epithelia in your lower and upper respiratory tract is not going to produce this alpha-2,6 sialic acid galactose linkage. But the scientists were able to infect them with the influenza virus. So what that does mean? And it was found that instead of this alpha-2,6 sialic acid linkage, they express this alpha-2,3 linkage, which is different. And this gene does not cause the expression of this gene, is not responsible for the expression or production of this alpha-2,3 sialic acid galactose linkage. And the virus can use this to enter the cells. And that we have seen in the avian strain as well, that there is a difference between these alpha-2,6 and alpha-2,3 linkages. So as you can see, that the cell culture gives you one result, and you do not have the same output from an in vivo or an animal study or the human beings. So these are some of the limitations. On the other hand, you can see that the animal models are used, and unfortunately that we say that the mice lie and the monkeys exaggerate. What it means that it's funny, but the thing is that what you do in the mice or the ferrets or the monkeys, it is not exactly the same as in the human. And it is very difficult to conduct this experiment to see that. There are only a few examples like influenza and rhinovirus, common cold, that human beings are deliberately infected by those for the study purposes, but they are extremely expensive to conduct because you have to give them huge amount of compensation due and insurance and all those kind of things, even with those viruses. But that is not a feasible or mainstream way that we study viruses. So how we do this, and I have written a detail of those. Those are self-explanatory. You can just go over that. And just a couple of them that you can just easily understand, that the mouse, you can just make the transgenics very easily. And this is, that you can take out a gene, you can put a gene, you have all these different type of technologies like a CRISPR to silence a gene. You have this CRE-LOXP technology. Just look it up. I'm sure that you have seen it somewhere else. And these can actually take out a gene or put a gene, and you can do these in the germline of these mouse easily to make this transgenic. So the mouse can synthesize human receptors, and now the virus can enter into them. But will it replicate in the cell? It could be susceptible, but may not be permissive. On the other hand, sometimes some of the virus which doesn't actually replicate in the mouse, you can put the whole viral genome. This is done with the hepatitis C virus. You put the whole hepatitis C virus genome into the mouse and see the pathogenicity, because it cannot be infected by the human hepatitis C viruses. And also individual viral gene, you can study the host response. These are mammals, so they will have similar kind of immune reaction, but not exact. So on the other hand, you can see that you can put clonal T cell receptor, immune mediator deletion. You can just take out one of the pathway which triggers the immune system. You can say, for example, that we have heard about this TLR 3, 5, 8, 9. All of these detect. You can just take the TLR out and then infect the virus and see that what happens. You can delete the immune cell. This is very commonly done. That commonly the mouse are mouse. You can just get the laboratory mouse, which are completely deleted out of B cells. They cannot produce B cells. Some of them are completely devoid of the T cells, and some of them, which we call usually the nude mice. That's a word that they do not have the skin or the fur on top. They do have the skin. They don't have the fur. That's why we call them the nude mice. And these are T cell and B cell deficient, both. They don't have any adaptive immune system. They only have this intrinsic and the intrinsic and the innate immune response. And all of these models are actually used to see the function of these viruses in the animal models. And we can see that if you overproduce an immune mediator, if you just overproduce an interferon alpha or a beta or gamma, what happens to the viral infection? So all these can be done in the animal models, but the problem is that it is not exactly what in the human beings are, and that's why we often say that as a fun, that mice lie and the monkeys exaggerate. All right, so after that, we are looking at some of the patterns of infection. And in the patterns of infection, you can see that acute infection is a rhinovirus, which means that it appearance of the symptom, and in the course of time, it just goes away. On the other hand, one of the confusion that happens is that what is a latent infection and what is a persistent infection. Now, this latent and persistent infection is very overlapping. So do not try to separate latent and persistent as well. Both is both, and I'm just going to explain that what it means. So you can see that all of these infection, if they are staying in the cell for a long time, the period of time they are in the cells, we call that as a latent infection. But the persistent infection, sometimes you can get rid of, but not always. So if they are there in the cells for the rest of their life and not doing anything, we call them latent. But again, persistent means that they are there all the time, but sometimes they can be asymptomatic, but there is viral replication. Sometimes they could be symptomatic, where the viral replication can cause pathogenicity. So let's take a look at a latent virus, a latent infection of herpes simplex. So you can see that, say, for example, cold sores. So there is an onset of cold sore, then it goes into the latency and again come back, and so on. And sometimes the virus may replicate without the appearance of the disease as well. You can see that there are some cells where it is being replicated, but you don't have the onset of the disease. That can happen in the latency as well. And usually most of us are infected with some kind of herpes simplex virus for life, and we cannot get rid of it. Say, for an example, if we have chicken pox, we have the varicella zoster in our sensory neuron for the rest of our life, and it is latent. On the other hand, here you can see that persistent asymptomatic infections, say for an example, LCMV, lymphocytic choriomeningitis virus, this JC virus, this is known as a John Cunningham virus from the patient was recovered. These are persistent asymptomatic in in the humans as well. So you get that, and they will be staying in your system for the rest of your life, but they may not cause any disease because your immune system can keep them in check for the rest of your life. On the other hand, you can see that some of these infection that human deficiency virus that are pathogenic, T lymphotrophic virus, even the measles virus, it can establish latency, and then at certain point of time, it can just come out and cause the disease as well. So this is a kind of like a chronic type of infection. That's what it is. Latency means that it's in there and constantly not doing any harm, just coming on and off periodically. But this period that when it is not causing anything, we call this one as a latency as well. Persistent infection can be cleared in some of the cases, not in the immunodeficiency virus because it's a retrovirus. That's a latent virus. Say, for example, the measles virus, you can just get rid of the latent, you can get rid of that. That's why it's persistent, but not the latent. But the latents are usually most of the time that that stays with you for the rest of your life, with or without any symptoms. So here we are looking at the varicella zoster, which causes a chickenpox, and see that how it happens. It can enter through the eyes or the mouth, nose, any mucous membranes, and then it will replicate the primary lymph node, and we call this as a primary viremia. So in this primary viremia, the virus actually will move into the liver, spleen, and other organs. And from there, and remember that the viremia is that when you have detectable virus in your blood as well. So through the blood and the tissue fluid, it will move into the skin. And now you have the appearance of the rash due to the immune response. And then what it does, that it will establish itself into the sensory ganglion. They will get into the axon, which are the end of these axons of these sensory ganglion are in your skin. And why they are there, remember that from if you just have taken anatomy and physiology, these sensory neurons end up on your skin, and they are touch receptor. They are mechanoreceptors, like touch receptors, can sense temperature, pressure, heat and cold. All these tactile functions are by these sensory neurons, and they enter onto their this is your receiving part, and these are does have a single axon. Remember that they really do not have an end part or a dendrite. We call this that they are just one long axon, and the cell body is right here, which is the sensory ganglion, and we call these as a unipolar. These are unipolar neuronal cell, and the viruses enter to the sensory neurons on this end, which are basically in the skin, and then they will move into the sensory ganglia, which is a collection of these individual sensory neurons, which is just close to the spinal cord, and they will stay in there for the rest of their life. They can be reactivated to cause the shingles, and it has been shown that some of the cases, stress actually do reactivate this shingles. And as you can imagine that usually the shingles is way more painful in there, and the shingles happens, a rash happens in the shingles, but it doesn't happen to the entire body. It just only happens in a specific band of your body. It could be a portion of your face or a torso, anywhere where the reactivation of the varicella zoster virus actually happens. Say, for an example, if this middle neuron actually got reactivated, and suppose this middle neuron actually supply to the skin, which is in your upper torso, a certain area. We call this as a particular area or a band of your skin is supplied by that, and you get the rash only in that region. but this rash is much more painful, and there is a vaccine also. You know that. So this is basically that you have the primary acute infection, and then it establishes the latency in the sensory neuron where it stays for the life. And for many of the most of the people, you may not get a shingles at all, but they will still be latent in there, and sometimes you can get the shingles at a later point of time. Another example, herpes simplex virus. There are many different types, and how it happens that they initially infect the epithelial cells. And after the initial replication, the same way that they can infect the sensory ganglia, right? Exactly the same way that varicella zoster. Remember that this varicella zoster is also a herpes virus, so it's the same like a same family of viruses. So after it get into the sensory neuron, it can just go into the sensory neuron body of the sensory neuron, and it can actually replicate right here. But most of the time, what happens, the immune surveillance cell actually can keep them in check in this position. But on the other hand, sometimes what happens that it can move from here, from the sensory ganglion, towards the central axon to the CNS. So you can get a CNS, central nervous system infection as well, if your immune surveillance cannot check that. There's another route, just a little bit difference in that sense, that this is the autonomic ganglion system. So here, so sensory ganglion is just one circuit. From the periphery of your skin, it supplies to the spinal cord. Whereas the autonomic ganglion, there are two circuits. You have one peripheral ganglion right here, peripheral neuron, sympathetic neuron, which supplies to a secondary neuron. This will connect to the spinal cord. If you just look up that the sympathetic neuron or autonomic nervous system, you will see that it is a two neuron system. So it's the same way, but principle is the same. It just gets infected, and the peripheral axon, through the peripheral axon, it can move into the cell body. Immunosurveillance can keep them in check, but the virus can be reactivated from here, and also it can move into the CNS. So when these viruses are actually reactivated, you can see that they can just move into your epithelia and replicate, and these can cause time to time your cold sore, fever sore, these kind of things in the herpes simplex virus infection. Here is another one. This is also a herpes virus. All of these are herpes virus, and what we are looking at is that all of these are establishing latency. Latency here, and latency here as well. For some of the EBVs or Epstein-Barr infection, some of them may not have any symptoms of the Epstein-Barr virus for the rest of their life. So this actually caused the infectious form, caused the infectious mononucleosis, which is commonly known as mono, that you all know. And the virus enters through the epithelial cells in the oropharynx, in the mouth, and that's the reason that they are called the kissing disease for that reason. And from there, they infect the resting B cells, and they move into the blood vessel. And at that time, these resting B cells, after getting infected, these B cells do produce some of these Epstein-Barr virus nuclear antigens. These are some very specific type of antigens which can cause these cells to transform sometimes into immortal cells. That is the important part. They can transform these B cells into immortal cells. That can happen in the cell culture as well. And sometimes that can lead to cancer as well. Not always, but sometimes. But they can make these B cells immortal. These cells produce these two membrane proteins, which is known as this LMP1 and LMP2. So these are the important two of these proteins which are produced in the on the membrane by these. Now, when these are actually produced, what happens, as these are the markers, some of the cytotoxic T cell and natural killer cells are going to kill them. And it clears most of them. But what happens, this is the thing, approximately 1 in 100,000 probably, they just can escape from these cytotoxic T cells or the NK cells. So that just a few of these T cells, which are the memory T cells, can stay, and they are now latently infected, and they only express this LMP2, not LMP1, only LMP2. And these are the latently infected resting memory cells can stay in your lymph node for forever. If they express both LMP1 and LMP2, the cytotoxic T cells can be activated, but just with the LMP2, it cannot get recognized by that. And these can be reactivated, which can produce the infectious virus, which can infect the peripheral cells and move into the saliva and can be infection. And this is your persistent infections. So you can see that there are several different ways that the first one is the sensory ganglion latency, sensory ganglion and autonomic ganglion latency. Here, the latency is in B cells in the immune system. So just the primary and persistent infection and latency in different types of cells. That's what it is. So again, remember that your persistence and latency, we are constantly using these words interchangeably, and don't try to fight these words. These are very interchangeably used. See that here we say the latently infected resting memory cells, but I'm saying primary and persistent infection. So just to make sure that they are almost similar and there's very little difference in between them. So let's take a look at some of the other viral virulence properties. These are influenced by the dose, route of infection, species, gender. You can see easily that. This is one of the important things that is very different than what you have probably learned in your microbiology. If you have seen in your microbiology course, you have probably heard about the lethal dose or the infectious dose 50%, LD50 or ID50. And you will see that particularly in microbiology textbook, it is always compared that, okay, all right, probably your Rocky Mountain fever or Ebola or some of the Lassa fever virus, they are extremely virulent. On the other hand, rhinovirus or influenza virus are very less virulent. And this is by their LD50 value. So for microbiology purposes, general microbiology, that is okay. But in the virological perspective, you cannot compare virulence of different viruses. You just simply cannot. You can do that only by the similar viruses, and the assays must be the same. And we are going to see that why we cannot tell that really the Ebola virus is more virulent compared to an influenza virus. Now, remember that here the virulence is the ability or cause the pathogenicity of the disease. Remember that the one thing is important. We are not looking at the mortality, which is the indication of an epidemiological study. So the virulence is not epidemiology. It is the degree of pathogenicity or in the cells that is happening. So it's very different. So if the LD50 values that you see in your microbiology class is related to the epidemiological outcome, that is correct. You can compare those epidemiological study, but you cannot use the word virulence in between the different viruses. Just make sure you understand that. And for the similar viruses, the assay must be the same as well. So we're going to see that what happens in this case. So here we are looking at the viral virulence, and here we have two types of poliovirus, type 1 and the type 2. And you can compare them. Both of them are picornavirus, very, very similar. But you can easily see that why the virulence varies. Here you have the days of post-infection, and here is the number of survivors. So here you can see that when it's infected by the type 1, you have almost all of them survive. Whereas if you just look at the type 2, you can see that by 10 days that every one of them is dead. So you can easily see that you can compare the virulence of this because both of them are the same type of virus, poliovirus type 1 and type 2. On the other hand, you can easily see that these are, say, Japanese encephalitis, yellow fever, West Nile, Langet, dengue fever virus. Now we are comparing different viruses. And we just said that we cannot compare different viruses, but why we are comparing that? Anyone can tell that why we are comparing these now? This is rational, picornaviruses, both of them. So just to have an idea, say all of these viruses are flaviviruses. These are all belong to the Flaviviridae family. They are very similar in their genomic organization, and the mode of infection and everything. So all of them are flaviviridae. It is not that one is these are all plus-stranded RNA viruses, not that one is a negative strand, one is a DNA strand, one is that. They are all flaviviridae family. So, but you can see that how the pathological lesions happens in there. And these are the different areas that you actually see that in the central nervous system. And you can easily see, just depending upon that the relative neurovirulence score, that which part of the brain that they are having the pathological lesions. You can see that the dengue virus actually almost don't have any, being the same flaviviridae family, don't have much pathological lesions in the brain. Whereas you can see that Japanese encephalitis virus, which definitely caused the encephalitis, does have a huge amount of lesions in the brain. So that's basically the idea. And what we mean by this is that the C is the cerebrum, then you have the brains, the B is the brainstem, and this is the S is the spinal cord, the different parts of the central nervous system where they are. There is a difference in between them. And you can see that how different is this. For the Langet virus, you can see the lesions in the spinal cord is the highest, whereas this one, the spinal cord and the brainstem is the highest. So you can see that just the neurovirulence in different area in the lesions vary among the same family of viruses significantly as well. So next we are looking at the viral, also depends upon the route of inoculation, where you put your virus, and this is easily can be understood, that you can put this LCMV, which is a very, very good virus for modeling purpose. This is an arena virus, and lymphocytic choriomeningitis virus cause meningitis in the mouse. And if you put 100,000 PFU intraperitoneally, the mouse survives. You just put 1 PFU intracranially inside the brain, and there is a death of the mice. And the cause of death is immunopathological. And we are going to see that how this also in the later part. Interestingly, what happened that if you take an immunodeficient mice, that mice survives this persistent infection as well by this. So you can see that here, the virulence is dependent upon the immunopathology, and that we are going to see much more in the later part as well. So just an overview of that, the major goal of virology to identify the viral host genes and determine the virulence. And the virulence genes is usually identified by a mutation of the virus, and sometimes we are not lucky to find that mutation. Otherwise, it's really not very straightforward study. It just needs a lot of patients to study or find a viral virulence gene. You cannot just take one gene and just delete that, and that may be required for the replication. But whether it is a virulence gene or not, you cannot make that decision, because it is more complicated than that. So that's one of the things, that the virus that causes reduced or no disease in a specified system can tell us that what the viral virulence gene is, but it has to replicate. If you take a gene out or make a mutation, and if the virus doesn't replicate, you cannot measure the virulence. That's why it becomes very, very difficult. So here we are looking at that one of the experiment, that how it is that you have a wild type virus which grows in the cell culture. And you can see that the mutation leading to general defect in the reproduction right here. On the other hand, here is a mutation which is gene specifically required for the virulence. And how we actually look at this, that if you take the wild type, you just grow in the cell culture, you put it on the mice, and this is a neurovirulence, and this will kill the mice. On the other hand, you can have a mutation which lead to general defect in the reproduction. Of course, it will be attenuated. That's very, very logical. On the other hand, this basically mutation in a gene specifically required for virulence, where understand that the replication of the virus is as much as the wild type, but still the mutation at that gene cause poor reproduction in the animal, and it is attenuated. So this is the gene which is the virulence gene, not this one, because this is caused just the less reproduction of the virus, and of course it will be attenuated. a gene which is not compromising the viral replication, but still have an attenuated effect or reduction of the neurovirulence, that will be the neurovirulence. And this is the reason it's very difficult to identify this. So understand that very carefully. So what are these genes? And there are lots of different genes. Some of these virulent genes can be for replication, invasiveness, tropism, tissue tropism, where the virus can divide, how they can modify the host defense mechanism. We have seen that throughout the topic of these courses, that how they modify the RNA synthesis, translation, and all those things, enable the virus to spread to the host from one host to the other. And also the intrinsic cell killing effect. We have seen that, say, for example, HCV, directly apoptosis is caused by the HCV or intrinsic cell killing process as well. So these are some of the viral virulence gene overview of these. And we're going to look at that other than, since all these viral virulence, as we are saying that these are genes, that means the virus is going to produce, by the help of the cellular machinery, a protein. And that protein is the virulent protein, which will actually do this. But the virulence genes doesn't have to be a gene. It could be just a sequence in the viral nucleic acid. And here what we are looking at, that in the poliovirus, viral virulence determinants need not encode proteins. So what we are looking at, that this is a poliovirus, and there is a region, you all know that a 5' untranslated region, and this is basically the iris region. We have talked about that. And what you have, that there is a small area right here where you are looking at this. There are some small mutations which can reduce the neurovirulence, and this was a basis of the polio vaccine. So there are 3 strains of poliovirus 1, 2, and 3, and the Sabin vaccine, which is a classical polio vaccine, contain all the 3 strains, P1, which is a type 1, type 2, type 3. These are the 3 different poliovirus strains all mixed together. And there are some other mutations, but these are the ones which reduce the neurovirulence and make these strain as a vaccine strain. So what we are looking at, the P1, you have a 5' UTR. At the 480 position, it changes an A to G. 480, it changes an A to G in the P1 right here, A to G right here. This 480, this A to G. Then you have the P27 in 481, it's an A to G mutation right here. And this one right here is the P3 Sabin, a C right here. C is changed to an EU, uracil. And these just single nucleotide mutations can reduce the neurovirulence. But there was something that actually happened, and that is very interesting. There was a reversion of the P3 Sabin, and that happened a long time ago, that some of the children were actually inoculated by the vaccine and they developed polio, very unfortunately. And why this happened, that when the scientists analyzed the sequence of these Sabin vaccine, they found that there was some reversion occurred. And this is the many interesting thing, that this reversion does, I just wrote that, reversion does not mean that this C was changed to U and this U is changed back to C. No, somewhere else in the nucleic acid, there was another mutation which reverted this back to the neurovirulence. So now you can understand that why the vaccine development has to be tested so many times, because if particularly that's the reason that there is always a risk for the attenuated vaccine. If you're using a live vaccine, it has to be tested over and over so that this thing doesn't happen, that the reversion of this process where it can gain the neurovirulence. But you can see that the overall take-home message is that just in the non-coding region, some of these mutations can cause reduction in neurovirulence, which is the basis of the live attenuated strain of the poliovirus vaccine. So here we are looking at that mutation, just an overview of that, very simple experiment. So you have this 2 different viruses, whereas this is the mutation at the base 472. If you have this EU right here, you can easily see easily see the neurovirulence of these two different viruses. You can see that with the seed right here, this one, this is the days of post-infection, and this is the log plaque forming unit. You can see right here, this is the virus with the original seed. And you can see the LD50 dose right here is 9 into 10 to the power 3. On the other hand, you can see that right here, if you just replace that C with an U, how the dosage of the LD50 dose has been reduced, and it is greater than 2 into 10 to the power 7. So you can see that you have from 10 to the power 3, the LD50 dose went up so much. So for the vaccine amount, this should not cause any neurovirulence or cause any disease as such. So just one mutation can cause this difference in the mouse brain. You can see that from direct evidences. So other than that, some of the gene products that modify the host defense and what they do that they can induce apoptosis. We have seen that autophagy, and there are some intrinsic proteins as well that we have seen this HIV ApoBac3G, which is of course antagonized by the VIP protein of the HIV. But the ApoBac3G is an intrinsic protein which is designed to make hypermutated retrovirus in retroviruses as well, and in the human genome for a long time. HSV ICP35 4.5 is also another protein which also is an immune modulator. And along with that, many of the viruses actually produce these virokines and the viroceptor. And usually these are produced by the large DNA viruses because they do have these large amount of genes which can code these. So what are these? These can produce the cytokines or the receptors for the cytokines. And these receptors are soluble decoy, and the real cytokines released by your cell can be bound to these viroceptors, and the antiviral effect of those cytokines will be reduced. And then you can have complement binding protein. We have seen that in the spice. Remember that in the variola virus, if you just look at that particular complement slide in your innate immunity slide, it can also modify lots of MHC1 and 2 pathways. We have talked about that in the last section as well. And often not required for the growth in the cell culture as well. So that's one of the problem that we have, that the cell culture sometimes do, or most of the times do not have an immune response. And that's one of the reason also is difficult to study these products which can modify the host defense mechanism. So here is also a viral virulence gene we are looking at, this gamma herpes virus 68. This is an M3 gene. So this codes for a chemokine receptor, and see that what happens. The wild type mice is the blue one, and right here is the PFU. And if you just look at the, carefully look at the result, that we have, this is the percentage of survival, and you can think about that as an LD50. And if you just look at this virus right here, you can think that this is the LD50 value right here. So if you look at the LD50 value, see that this is the PFU. So see that this is in between 1 and 10. So approximately say 5 is the 5 PFU approximately. Just visually is the LD50 value of these viruses. So now what has been done, that you basically you make a virus where you delete that M3 gene, which is responsible for this chemokine receptor, so which is this one. Now let's see that where is the LD50. If you just look at the 50% LD50 right here, you can see that it is in between 100 and 1,000, and it is not exactly in the middle, so maybe around say 300 or 400, but you can see that from 50 to how much increase 300, 400 has been done. Now, just to check that this is really a virulence gene, this is called the delta M3 marker rescue, means that first the virus was deleted from the herpes virus gene, herpes virus genome, and then the gene was put back in. And when the gene was put back in, which is this is an old term called marker rescue, and you can see that the red curve right here has the LD50 value of exactly like the wild type. So you got the virulence back. So this is one of the example that how this can be determined. So here is also another example of viral protein can be toxic. One of them is a rotavirus, which caused the gastroenteritis or stomach flu. There is a protein called NSP4. What it does that it inhibits the sodium glucose aluminum cotransporter. Why this is important? Because this actually helps in the water reabsorption from the intestine. So that is required. So how this happens? Sodium glucose cotransporter, by the help of the sodium, which is more in the intestine, as the sodium moves down the concentration gradient, it brings in glucose as well in the cell. You have learned this in cell biology. So in this process, what happens? The amount of sodium in the intestine goes down, but the amount of sodium in the cell increases, and the cell actually also, which is not shown, get chloride from the environment. So for the electroneutrality, you have Na plus more and you get Cl minus in the cell. And as the Cl minus comes into the cell, you have Na plus and Cl minus, which is basically the salt amount in the cell increases. That becomes a hypertonic environment and it will absorb water from the intestine. This is one way that it blocks that. So if you do not have sodium coming in, no chloride coming in, no water coming in. There is another way that NSP4 also can signal the phospholipase C, which will increase the calcium inside the cell. And the cell does not like calcium. So what happens, this calcium will be pumped back into the lumen. And as calcium is pumped back into the lumen, the plus charges are increasing. Chloride, on the other hand, what will happen that it will increase also the chloride amount. And that will also cause the water to leak out from the cell to the intestine. So that's basically what happens. So see that two different methods. This is blocking the water movement into the cell. And this pathway is promoting water movement from the cell to the outside. Because remember that here what happens, carefully remember, understand the experiment. Sodium comes in, Na plus goes up. Chloride Cl minus goes up. More salt in the cell. That is the reabsorption. That is blocked. Whereas in this process, calcium is released out of the cell. Due to that, chloride minus, just to negate that 2 plus, the plus charges, chloride will be secreted out of the cell. So now you have this calcium 2 plus and the chloride that will attract more sodium outside into the cell. That will take out water from the cell to the intestine, which will cause that gastroenteritis or the diarrhea. So that's basically the enterotoxin NSP4 function. So we are looking at some of the cell injuries that what can happen in these. So there are lots of cytolytic viruses which can cause apoptosis, necrosis, and pyroptosis, just a different way. This is a programmed cell death by the caspase. Necrosis is a direct killing by some of the toxic substances, killing by the cell. Pyroptosis is when the cells die due to the inflammation. Sometimes there are some viroporins which cause the pore formation in membranes. Influenza virus M2, Picorna 2b, HCV P7. All of these actually make holes in the cell membrane, and that cause the cell death. And we have extensively seen how the host protein and RNA synthesis is blocked. And that all of these can lead to loss of membrane integrity, leakage of enzyme from lysosome, cytoplasmic degradation. And also you can see right here, this is a picture that we have seen many times. This is directly cytopathic effect that you can see here. You can see this giant synthesia formation, which is seen in the parainfluenza virus as well, which are the viral factory. So these are the different types of cell injury that is shown by these viruses. The next part that we are going to talk about is the immunopathology. And this is the too much of a good thing that can kill us. And unfortunately, what we are seeing that in the SARS-CoV-2, this immunopathology is the reason for this number of death. And what are these? So this is just an overview of that. What does this mean? All of these are due to your immune function. These are the consequences of your immune response. These are non-cytopathic virus. The virus is not killing but the reaction against the virus, your own immune system is actually killing you. So that's the sad part, that happens in immunopathology. So what are these? We are looking at the cytokine storm in the SARS-CoV-2, CD8 T cell mediated cell killing that can happen in these viral infections. These are very straightforward. CD4 helper T cell mediated that can happen in measles virus, herpes simplex virus, immunosuppression that we are going to see that. And CD2 Th2 mediated, this is also CD4 helper 2, but Th2 respiratory syncytial virus, RSV. Also, the antibody can cause immunopathology that we are going to see for the dengue virus. So we're looking at some of the examples. So here we are looking at the signaling this is a print mistake, this will be a delayed IFN signaling can induce killer cytokine storm. So here this happens in the SARS and at the SARS-CoV-2, that what we have seen. So first we are looking at this, and then we will compare the other two. So in the bottom one, which is the red box, you can see that as the SARS infection happened, CoV titer, immediately from the very beginning, there is no interferon signaling. So the virus is good enough to delay this IFN1 response. And this is done by a protein, if you remember that, the NSP1. This is at the very N-terminal end of the genome. And this NSP1 causes this delayed IFN response. So now what happens, that ultimately, when the virus is actually at the peak, almost at the peak, at that time, the IFN response happens, not at the beginning. And that time, suddenly your immune system wakes up and see this much of virus and this much of IFN, and this will cause a huge amount of cytokine release. And this blast of the cytokine release is known as the cytokine storm, and that is the lung immunopathology, and a lethal pneumonia is caused by this. See that what happens in other cases. If you have an early IFN response after the infection, you can see that there is a regulated inflammation and mild clinical disease. If you don't even have, this was experimented in mouse with the original SARS-CoV, SARS-CoV, which is a classic SARS of the 2002, not the SARS, that even if you make a mouse which doesn't have any IFN signaling, no IFN response, that doesn't even kill these mice. No lung immunopathology, mild clinical disease. You can your other parts of your cytokine responses can clear the viruses. But here you can clearly see that the delayed IFN response caused these cytokine storm. And this is kind of a mind-boggling from the recent paper, and you do not have to look at individual names or anything like that. Just look at some of the data that what this cytokine storm means and how mind-boggling is this. So basically what is being shown here, there are some of the other factors, but the green ones are the pro-inflammatory cytokines, red ones are the inflammatory cytokines, and these are the chemokines. The chemokines attract the immune cells to the site, and the pro-inflammatory cytokines activate the inflammatory cytokines, and the inflammatory cytokines will cause the inflammatory reaction. So all you have to see, I just did not do it for each one of them, that see that this brown arrow is where you have a healthy control, the first column right here, and see how down is this right here. You're looking at this basal level of this. But if you look at the ICU patient, which is the middle, and then non-ICU patient, look at the level compared to the basal level of each of the cytokine. And this is what we are calling about this cytokine storm. And if you look at all of these which are implicated by that, this is a recent paper, that what we mean by the cytokine storm is this: all these elevated amount of cytokines are responsible for the disease, severity of the disease in some of the patients where we see that this is lethal. So that's basically how the delayed IFN response is causing this, because suddenly the immune system wakes up and this burst of cytokines cause the immunopathology. So how this immunopathology looks like, you can see that overview of that. So in a moderate damage, if you see that the cells right here, these are the pneumocytes. This is a single alveolus. Alveoli is a plural. Alveolus is the air sac lined by the pneumocyte. And these does the gaseous exchange in between the capillary, which enwraps it. Right here is the capillary around the air sac. And in between this, you have the movement. And what happened that in between the capillary wall and this right here, the pneumocyte, there is some surfactants, and those surfactants actually help in the gaseous exchange in between this empty air sac as well as with the capillaries. So when you have this infection, these cells, which are the alveolar cells, type 2 alveolar pneumocytes, they will release this inflammatory signal. They will cause the cytokine release that will cause the vasodilation. That means the capillary around that alveolus will increase. As the vasodilation occurs, that's an inflammatory response, it will secrete more tissue fluid into that. This is as simple as that's when you have a mosquito bite, you just have a bump in there. This is due to the inflammatory response because you have more blood flow into that region that increase the amount of tissue fluid. This is the same thing. You have a vasodilation, you have more water coming into that area, and it fills up this alveolus partially with water. This is basically the pulmonary edema. Now you have a much less surface for the gaseous exchange, and that is the moderate damage. On the other hand, in a severe damage, this fluid accumulation completely fill up the lungs, and along with that, there will be scar tissues, which is that we call this hyaline membrane, because the cells are going to be killed, and that's going to form some of the scar tissues, which will bring in some of these fibroblasts to make these collagen fibers in these areas to repair those tissues. And those will be completely detrimental because gaseous exchange cannot happen through these scar tissues. And this is completely a protein-rich fluid that will cause basically the ARDS, or acute respiratory distress syndrome. There is no gaseous exchange possible in this condition. Just another rendition, if you take a cross-section of this, how it looks like in a normal type 1 and type 2 pneumocytes, you can easily see that they are all lined by this surfactant. And these surfactants help in the gaseous exchange in between these cells, type 1, 2 pneumocyte, and the empty space of the alveoli, alveolus, singular alveoli, plural. Now, when you have the infection, all these cells are going to secrete these cytokines like TNF alpha, IL-6, IL-1 beta, CCL2. Again, you do not have to remember the names, but a lot of different cytokines. These are going to attract all those immune reactions and the vasodilation, which will cause the water or fluid accumulation, are going to bring in the chemokines, are going to bring in the neutrophils, and it's going to fragment the cells. Some of the cells are going to be killed by the cytotoxic T lymphocyte, and these fragment or dead cells are going to fill up the protein fluid, and some of the areas are going to produce this hyaline membrane. This is the scar tissue by the fibroblast, and all of these will basically block the gaseous exchange. And you can see the surfactants, which is right here, that is reduced, that is even more reduced in this condition, ultimately leading to respiratory failure in the SARS-CoV-2. We are going to see more detail of that in the special topic, though. Some of them, we are going to see that. All right, so after that, we are looking at, other than the cytokine storm, that how the CD8 cells actually cause this problem. Here you can see the lymphocytic choriomeningitis virus. If you just inject intracranially, this is lethal choriomeningitis. On the other hand, if you take a mice which doesn't have any T cells, or you suppress the T cell function or the cytotoxic T cells, you can there is a persistent infection. Now, if the T cell deficient mice, if you just can adoptive immunization, that means you put back the T cells into this mice, the mice actually dies. So you can easily see that a CD8, your cytotoxic own T cells, are responsible for the lethality of this disease, or the LCMV. So what are these lesions associated with CD4 lymphocyte? Elaborate more cytokine than CD8 cells, and recruit, activate many non-specific effector cells Most recruited cells are neutrophil and mononuclear cells, which are protective but cause tissue damage. This is what we actually seeing in the previous chapter right here. You can see that this neutrophil has been called, and that's what it is, that these neutrophils, you can see that the chemokine release and the cytokine release, this is a neutrophil from the blood, and from the blood capillary, they are infiltrating, and this is the process called diapedesis, by which they leave the capillary and move into the lung tissue. And all these neutrophils are going to produce chemicals which are detrimental for the cell, and it will cause the tissue damage and the killing process. So that has been elaborated here. And the immunopathology caused by release of proteases, reactive radicals, many of these, and we have detailed that, and also the cytokines like TNF alpha. So all of these we have seen that happens in the SARS as well, in the SARS-CoV-2 in case of the cytokine storms. Here is one thing which is associated with CD4 plus T cells. And this is a condition which we call as a herpestromal keratitis. And this happens in the developed country. And this is due to the entire pathology of the CD4 Th1 type cells. So what happens here, that here you are looking at the cross-section of the cornea, and you can see that this right here, this cornea is completely normal cornea. Here you can see that these herpes virus replicate in these corneal cells. What happens that these cells release the cytokines, and they will attract these CD4 cells. So you can see that these are the endothelia right here. These are all these nucleus that you are seeing in the stroma portion. They are right there, and they are underneath the epithelial layer. So what you are seeing that the CD4 T cells, they are not even in the epithelia, but they are underneath in the stroma. But all these release these cytokines, which actually is detrimental and make the damage to this top layer. So the cytokines are released from that layer or the stromal layer, travel all the way up and cause the keratitis. So this is another which is caused by the CD4 T cells. This is another thing that when you have this, that TLR3 is one of the way that we can sense the virus, and West Nile plus strand RNA virus that is detected by the TLR3. So in this experiment, what was done, that in the mouse, it was infected by the West Nile virus, and there was an antibody, a stain was injected that is called Evans blue. And that Evans blue is going to give us the idea of the blood-brain barrier. So there is always a blood-brain barrier, and it is not easy for the viruses to cross that to have the brain infection. They are remain in the blood, but they cannot cross that barrier. So what happens that TNF alpha in some of the viral infection, when it increases, it compromises the blood-brain barrier. So what happens that when you have the wild type mouse infected by the West Nile virus, by day 3, you can see that the Evans blue stain that has been used, that has moved into the brain, which means that the WNV caused the blood-brain barrier to be compromised. That's the reason that the dye moved into the brain. But what happens that if you take a mice, which is TLR3 minus, means that it is a complete deletion of the TLR3. These mice don't have any TLR3. You infect that mice with the West Nile virus and see that what happens. There is, after 3 days, there is no permeability or there is no compromise of the blood-brain barrier, and the dye Evans blue did not reach the brain. So here you can see that the detection of the virus TLR3 and the TNF alpha itself, which is a cytokine, is causing the pathology. So that is immunopathology. Here is also another example of the poxes and the rashes. And we see that in the characteristic rash, measles, smallpox, Th1 macrophage is activated in the infection, homing on the infected foci of the skin. And these release this IL-2 IFN, which cause these increase in permeability and influx of T cells. Also, very interestingly, the dengue fever is a problem in many of these Asian countries, Caribbean countries, lots of infection. And this caused an antibody-mediated problem. So the primary infection is sometimes asymptomatic, sometimes febrile illness, and by 7-10 days, it is self-limiting and patients recover. But what happens that 1 in 14,000 primary infections that can cause an hemorrhagic fever or life-threatening disease, which cause the internal bleeding, leads to fatal dengue shock. But when you have the first dengue infection, you have antibodies to the viruses made, and there are 4 serotypes, dengue 1, 2, 3, 4. There is no cross protection in between. So you can be infected by dengue 1, then dengue 2, later dengue 3. It can happen. You will be infected, but that there is a problem in that step. What happens that when you are first time infected by the dengue type 1, your monocytes, these are the circulating monocyte in your blood, do have this Fc gamma receptor on them. And there are some heterotypic antibody which is circulating from your previous infection. And they bind to these new type non-specifically. They are not very specific to that, but just randomly bind. But what happens that these randomly bound antibodies to the new one, this is not enough to neutralize the virus. But just random binding cause these dengue virus to bind to this Fc gamma receptor of the monocyte, and the monocytes get infected. So what happens that this enhanced infection of the monocyte increase the viral load, and that can lead to 1 in 90 or 1 in 50 in subsequent infection, hemorrhagic fever and shock syndrome. In normally it is 1 in 14,000 in the in first time infection, but in a second or third time infection, the amount goes up to 1 in 90 or 1 in 50. And this is caused by antibody mediated response in the dengue fever. Then we're looking at the last one is the global suppression by the measles virus, and how we are going to see that how the immunosuppression is caused by a measles virus specifically. And this is due to the perturbation of the cytokine homeostasis by the intracellular signaling, or the viral proteins acting as a viroceptor, or the virokines that we talked about. So here what we are looking at, this is a tuberculin test, and you all know that how the TB test is administered. You take a tuberculin and you just put it subcutaneously, and after a day, 2, you are going to get a bump, and that is measured. And usually if it is more than 10 millimeter, it is positive. If it is less than that, then it's not positive, but you are going to get a bump. But what happens that if a patient is infected with the measles virus, see that without the measles virus, a patient which is not positive for the tuberculin or TB, just having approximately a 7 millimeter in duration, 7 millimeter bump. See that what happens that when you have the measles, there is no inflammatory reaction to produce this bump. The bump is gone. Then it regains against the after 2, 3, 4 weeks later, gradually over the period of time. So this is the classic example that how your inflammatory response is being suppressed by the measles virus infection. And how it does that, I will just take a second. The last slide, that we have seen this, that how the measles virus skew the production of the cytokines. This is from the slide that you have seen from the previous, I just mentioned, this is the slide number 22 from the previous topic, that the first, what happens that the virus, while it's infecting these detected by the antigen presenting cells, should produce IL-12 to activate the CD4 plus Th1 cells. And this is the first response. But measles virus stops this IL-12 production. On the other hand, activate these antigen presenting cells. This is needed later in the infection. At the initial stage of infection, there is no function of these CTL or the cytotoxic T cells activation through the Th2. So this skewing of these IL-12, antagonizing of this IL-12 production basically cause the immunosuppression. And these are some of the examples that you can see. The measles, rubella is called the German measles, HIV, which cause the cells monocyte and dendritic cells, thymine epithelial cells, rubella also lymphoid cells, and in HIV, you can see the CD4 T cells and the monocytes are infected. And these are all the immunosuppression that cause the pathology. So we will stop right here.